sorry, go ahead, yeah.
Okay. All right. Well, thank you for coming this morning. And so as I said, I'm going to start off with some overlap with Randall's talk. And in fact, as a chemical engineer, I'm really interested in what physicists will think about some of the structures we're seeing, to get your input on that. The work has been a collaborative effort, graduate students and postdocs here at Davis with Jarek Majewski at Los Alamos National Lab. And so with that, what do I mean when I say just a two-dimensional single layer? Is that we're going to be looking at biological membranes that are on solid supports in this case. And I'm going to start off with talking about some simple systems of one component, a little bit more complicated mixtures, as well as systems that are on cushions to drive this into some biosensing applications. And what's kind of unique about this is we have new capabilities to really precisely characterize the structure of these two-dimensional films, okay? Just this single bilayer of molecules. And that's really led to some new insight in how we understand their structure. And of course, we're always trying to relate this back to also understanding how true biological cell membranes operate, the fundamental principles that govern their structure and behavior. In the past, when we talk about scattering experiments to do structural characterization, especially with X-rays, the majority of work was done on monolayer systems. Okay, because of X-rays' poor penetrating power through materials, usually you had a single leaflet of the membrane that was studied. And then, as well, there were studies on multi-layers on a single surface where you had thousands of layers of membrane that you could characterize your structure, as well as enclosed vesicles, multilayer vesicles. And what's unique about the recent work is that we've been able to turn this characterization power onto a single supported bilayer. Okay. What we're going to use for the characterization are both X-rays and neutrons, and we do two different geometries. In the first case that I'll talk about, I'll share some results of reflectivity measurements. And in reflectivity, you're coming in at low angles and you're looking at elastically scattered neutrons or photons. Okay? And it gives you a pretty precise measurement of the density distribution normal to the support. Okay? And so you can get things like layer thickness, roughnesses, that density distribution, as I said, for these sorts of measurements. And these have been done for maybe, I guess, 20 years, okay, on these sorts of systems. Neutrons in bulk water and X-rays at the air-water interface. As I said, I gives you structure in the, normal to the plane of the membrane. But what's unique now is the ability to do grazing incidence diffraction. Okay, on a single supported membrane. And if you can do diffraction, what that enables you to actually characterize is not only the unit cell dimensions, okay, but as well as how ordered molecules are. This is looking at lipid tails within the plane, as well as things like molecular tilt. Are they tilted to the next nearest neighbor, so forth? What is that tilt angle? As well as what is the diffracting entity? How long is it? Is the entire lipid leaflet diffracting or just a single tail? Okay, so you get very precise information if you do diffraction. And as I'll show you, we were successful in doing that. And that's going to give us a very precise measurement of the structure within the plane of the membrane. And this also correlates with some of the huge interest that's been generated recently on what is the lateral organization within a membrane. There's questions about whether or not you have domains that are very small domains called lipid rafts that have, say, 50 to maybe 100, or I guess 50 nanometers sort of sizes, 20 to 50 nanometers in sizes. And this is an appropriate measure using grazing incidence diffraction to actually access it.
Just for clarification, the Q vector is now is now parallel to the surface?
So now we drop below, so we're dropping below the critical angle now, in this case. So we have an evanescent wave, and we're, instead of looking at the reflected geometry, reflected intensity, we're swinging out and looking at in-plane diffraction. So if we have diffraction. Okay. And I'll show some image plates of what that intensity looks like. So these are pretty clear. So just some data to start off, and these are the first measurements we did maybe, I guess, 5 years ago, 6 years, 5 years ago, I guess. On the left-hand panel, I show reflectivity, so that's the reflective intensity over the incident intensity, and you can see if you're below the critical angle, you get perfect reflection. But it rapidly drops off, power law 4, and this is the momentum transfer vector in the z direction, and you can also think of this as sort of a reciprocal space. And so this periodicity is due to the layer thickness, and the intensity of these peaks is due to that contrast, due to the density distribution of the layers from the interface. And what's shown in blue is X-ray scatter, X-ray reflectivity, and what's shown in red is the more traditional measurements that we're able to do with neutrons. And you can see even 5 years ago, the sort of information we got was much richer. The error bars are actually shown here for the X-rays; they're just below the data points. And we go out twice as far. We're almost out to 1 reciprocal angstrom now. Okay? You can convert this reflectivity profile. You actually model it using slabs of various thicknesses to account for head groups, tail region, head group. There's a letter, and here's our support. And you can map out what the density distribution is of that membrane, and it corresponds very well between X-rays and neutrons. Here's the unsmeared, because they have thermal fluctuations and so forth. Here's the unsmeared scattering intensity. It matches very well. And you can get very precise characterization of the layering out of plane. And I don't want to go into that too much, other than to say with the most commonly studied lipids or phosphatidylcholine head groups, you get a very thin water cushion. It's 4 to 5 angstroms of strength between the bilayer and the support underneath. And that, we'll discuss some of that as I go on. So the advantages of these X-ray measurements, you crank up the energy of the photons, and you can actually penetrate through 1 centimeter thick layer of water, and you can probe the structure here of this bilayer that's on support. Benefits are these measurements are extremely rapid. It opens up dynamic measurements. It takes about 10 minutes to do a reflectivity scan. When I talk about diffraction, we're doing those in 1 second. So you could actually look at dynamic changes in the bilayer structure, say with protein binding, or changing temperature, or electrolyte concentration, or whatever you want to do. You can do that dynamically. If you compare it to neutrons, a reflectivity scan here takes about 3 hours, and you're intensity limited. So in this sort of system, you couldn't really do diffraction at this time. Maybe with higher intensity sources, you might get that. The downside is you can have beam damage with X-rays because they are interacting and depositing more energy in the system, but be careful to avoid that. Okay, let's see. So you can look at things like self-organization. You can mix lipids that will phase separate. In this case, it's a C12 chain that's mixed with a C18 chain. Those will phase separate because of the very large differences in the chain lengths. And from the X-ray scattering data, we can actually tell that in spite of AFM measurements, what our measurements revealed was that the longer chain lipid goes against the solid support, and the outer leaflet that's against both water is enriched in the short chain. Okay? This was in contrast to the AFM measurements. Those demonstrated that these longer chain lipids were coupled across the membrane, and so that you have this height difference of 18 angstroms, which would only be consistent with DSPC, the 18 two leaflets together, and then a lower region where you have the shorter chains. Okay? But what it comes down to is what is equilibrium in these systems. If you fabricate this system and have it quenched, so that you're rapidly dropping the temperature below the melting point, you can see these sorts of coupled domains across the leaflets. But if you more gradually cool the system and give it some thermal annealing, you end up with the system that we saw. And this has been confirmed using neutron scattering measurements with deuterated lipids, where you can tell precisely which lipid is going into which leaflet. Okay, so I think with all these systems, there's always a question of what is equilibrium, and whether or not we're even in equilibrium with any of the systems is another question. Okay.
There's this phase diagram, isn't it, as a function of color?
In lipids, yes.
Various.
We'll get to some of the tilt. The tilt usually in PCs is fairly established. It's about 30 degrees consistently with phosphatic overheads. That's right.
But I mean, there's a phase diagram.
Yeah, you have
which phase?
L alpha. These are all alpha.
So you're going to be one particular phase.
That's what we're pushing. You can also, these are a little bit later measurements. We've gotten better. We're out to 0.8 reciprocal angstroms here. This is a bilayer shown in red. And then a more traditional measurement of a single leaflet at the air-water interface shown in black. Okay. If you look at the structures of those, how different are our measurements of monolayers? Oh, sorry. What is there? So we convert this into our scattering density profile. Here's the monolayer data, right here. Okay. And it's just a mirror image over there. And then in red is the structure that we measured for the supported bilayer. So it maps up quite well to the monolayer case. The resolution of the measurements is so high you can actually tell exactly how much water you have in between the support and the bilayer. It's slightly more water than what's associated here with the outer leaflet of the bilayer against bulk water. 6.5 angstroms per lipid in the inner leaflet and 4.5 in the outer. That matches very well to multi-layer measurements of the amount of water that you get if you have, thousands of bilayers stacked. If you look at their structure and how much water hydration you have. The areas per lipid, I don't know if this is getting too low, but it also matches up very well with both membrane measurements as well. So it seems like we've got a system that is reasonably well equilibrated in this case. There is variability, though, in these systems, depending on how we fabricate the membrane. We can do things like vesicle fusion to create a supported membrane, or we can laminar logic deposit individual leaflets. And this is again, this scalar density profile. Here's the head groups support. And these ribbons are actually showing the variability depending on how we fabricate the bilayer, what sort of structures we get. And it also points out why many groups have differences in behavior of the supported bilayer. It's depending on how you make it and what happened that day. You have variability in the structure. You can also look at the impact of different head groups. So this is a phosphatidyl ethanolamine that instead of having methyls has 3 terminal hydrogens. And in this case, there is no distinct water cushion for an ethanolamine head group. It's known to be less hydrated, and we actually don't even need a discrete water layer there, which is sort of interesting as well, and against what people have previously thought. You can map out some of these interactions of what you'd expect, and depending on time, maybe I'll glance over this pretty quickly, but in these lipids are zwitterionic, so they're overall neutral, but they are charged. In low salt concentrations, you actually have a fairly appreciable long-range electrostatic interaction. That's shown in dashed blue. And at short range, you have the van der Waals interaction between the support and the bilayer. And then in black here is the total overall interaction. And at low salt concentrations, electrostatics is actually dominant until you get to very short distances, below a few angstroms from contact. And you have a long-range attraction which stabilizes the bilayer on the support. These could be robust for weeks. Okay.
Question: is it the distance measured in the transverse direction? These are long things, so they're not point objects, so I don't know what you mean by distance.
Distance between the membrane and the support.
Okay.
Just measuring that interaction. And I'll show you something later where we have polymers in there where we can modify some of these interactions.
This charge is the one on the membrane?
So the electrostatic interaction is due to the quartz is canonically about a -50 millivolt potential. zero potential, okay, you still have a very long range electrostatic interaction. That will get cut down dramatically if you increase the time, or decrease the time. Right? So you add electrolytes in there, and you're going to snip down that
Right.
electrostatic.
My question is, can we differentiate? There is a static charge on the membrane, and there is a double layer from the water.
There's
From the
no charge on the membrane. The membrane has zero.
so this is all induced by the this is now in the water, or you have
It's in
electrolytes?
water.
So if you add electrolytes, this double layer changes the charge adjacent to the surface of the membrane, just underneath, will change this whole graph.
The range and the contributions of the electrostatic interaction will decrease.
Yes.
Yes. But this assumes even in the presence of salt, you don't have to have an effective surface charge on the membrane for it to be an attractive interaction. And that's solely because, of course we'll have something in between, we'll have target regulation per se, right, as well. But this is assuming constant connection with the electrostatic. In that case, with just having asymmetric charges, you have an attractive electrostatic interaction. I can also assume the membrane, I can give another example. I have a membrane that has a negative 100 millivolt charge. So both the substrate and the membrane are negatively charged. I will still have an attractive interaction at low salt concentrations because of the asymmetry.
So this is kind of free charge to move parallel to the membrane.
Yes.
And so a few years ago, I had done this study here. There is a collective mode, a plasma two-dimensional mode, which has a definite dispersion, square of K, along the membrane. And I wonder whether you can see if the square is in angle scattering.
We
Of the
can
definite mode.
see where it's sitting, right? So we can work out what the energy minimum is based on the separation between the membrane and the support. We know the van der Waals interaction pretty well. Okay. So then we can back out what we have to have for the electrostatic interaction. You can then play around with adding small amounts of charge.
Yes.
And then shift that around. And if you could do that systematically, then you could precisely tell. You can also map up some of this by having vesicles of the same composition. Make giant vesicles and put those in an electric field and back out what their effective zeta potential is based on their displacement in the field. If you have these lipids and you put those in that sort of measurement, you would see them move. They're effectively not charged.
So, this charge can move along the interface, and it has a definite wave frequency as a function of the wavelength.
Right.
And so the prediction, if you have a double layer, it's different prediction because you have two modes. If you have a single Lipid Monolayer, then you're getting different dispersion with the wave number. So that's square root. But, yeah. So I have a definite prediction for this kinds of collective modes. Nobody
Notice
been able
that
to look at it. Yeah.
Well, I'd be interested in talking to you about that. Okay. So where are we at now? We have a lot of data on the impact of chain length, head group. We're playing around with charge in the membrane now to try and systematically map some of these aspects. But I don't really want to spend too much time on that because I wanted to tell you about the fraction. So I think the most interesting questions we were interested in, first off, was this idea of trying to map onto this huge question of the presence of rafts and membranes. So fluctuating domains that assemble, that carry out a function and change protein conformations within the membrane, but that also disassemble to change the protein function. And I haven't done the search recently, but I did the search, I think, in like 2008, at the beginning of 2008, and this idea had come out maybe 10 years previously, and there's at least 6,000 publications on lipid rafts that have happened. Okay. But they've proven extremely difficult to quantify and measure. It could be liquid ordered, but it should still be fluid. Okay, so there's a I guess a quantification there on the impact of diffraction measurements. But what ends up being interesting is, as I said, the precise information about things like leaflets being coupled, how crystalline are the regions, the size of these crystallized domains, and so forth, and we can get that all out in diffraction. And so here's what it sort of looks like. This is in light of that one question of the geometry. So you drop down below the critical angle, and this is the quartz surface, here's the bulk water, and here's our bilayer that's sitting on the surface, and this is the diffractive intensity. And this is for a DPBE bilayer, and I'm going to show you the diffraction and how it looks from a monolayer. And just from this, there's no splitting, it's a single peak. That suggests that the molecules are not tilted and that we have hexagonal packing. And when you compare that to monolayers, so here's the bilayer diffraction on the bottom in red, okay? And here's the diffraction for monolayers at the air-water interface at different surface pressures. And so this sort of hexagonal packing is consistent with this type of packing of a monolayer, although we have less order and we have a slight shift to a larger area from the molecule in the region that's diffracting. But we can still say we have a hexagonal unit cell and it matches up quite well to the monolayer case. The splitting that you get in the diffractive intensity that happens with monolayers is due to the fact that as you decrease the surface pressure, you're giving more area for the molecules are starting to tilt over. Okay? So you get diffraction in splitting. So these are Bragg peaks, and as I said, you can also map out the size of these diffractive domains. Effectively in the bilayer, it's about 200 angstroms compared to a monolayer being 600 angstroms across. Okay? You can also I didn't say this either. This is integrating the intensity in the z direction. If we integrate the intensity in the qxy direction, so here we have it. These are called Bragg rods. And then, and again, I have the monolayer data on top, the bilayer data on the bottom, and you can see that the diffraction of a monolayer at this tight packing that's hexagonal looks very similar to the bilayer data. And in fact, 1 over the full width half maximal here is consistent with the diffracting length, okay, normal to the interface, and you can see that bilayer is diffracting like separate monolayers, okay? And so that length is 21 angstroms, that's the length of the DPBE molecule, the hydrocarbon tail switch shaft. Now this is different when we go to DPBC. Okay, in DPBC, here's diffraction from DPBC, just a change in the head group. This is monolayer data on the top, bilayer data on the bottom, and that's what the actual diffractive intensity looks like. We look at Bragg peaks now. It maps out pretty well. There's some changes in the position of the diffraction, so there's some shifts in the area of the molecule and things, but it maps on pretty well to about 40 millinewtons per meter. If we deposit the bilayers by Langmuir Blodgett, Langmuir Shaper deposition, we get better structured, better ordered bilayers, but still with vesicles, it still looks about the same. What's interesting, though, is that when you go to looking at the Bragg rods, we have this large change in the diffractive intensity off horizon compared to on horizon, compared to monolayers and bilayers. And what it actually ends up being is that there's no possibility of modeling the data with a closed packed system. So I plot this here as a closed packed system. We know that the molecules, if you model everything, are tilted about 30 degrees. Okay? And there's no way to have a closed packed system model this diffractive intensity. What you actually have to have is texture within the layer. So here's the tails have to be coupled. The diffractive intensity corresponds to about 42 angstroms in length, so it can't just be a single leaflet that's diffracted. They have to be coupled and oriented like this. And they also have to have, they're tilted 30 degrees, but they have to have some variation azimuthally to actually whack down this on-horizon intensity and still have the large off-horizon intensity. That sort of azimuthal variation is sort of like a swirl. This is effectively the sort of tilt variation azimuthally of plus or minus 12.5 degrees that we have. Okay, so you can see it's a very subtle effect. But it maps onto the sort of swirls that are seen in symmetrical crystals. Okay. Now our length scale for our sort of positional order for our diffraction is 200 angstroms, and here's 100 microns for the crystal. So it's sort of like a molecular precursor. But that sort of length scale is not inconsistent with what you see in fluorescence microscopy, as I'll show you in a second. What was really interesting we felt about this was that the bilayer is rapidly self-organizing. So we know in all cases that we only see coupled leaflets. And this isn't so hard to understand when you have vesicle fusion, where they have the ability to really, the lipids are very free to self-organize, and then that layer is carried down to the surface. But when we deposit leaflets separately, there's no reason for the orientation between the leaflets to be the same, and yet every time it's always coupled. And we do measurements frequently within an hour of depositing the system. So the whole, even though it's in the gel phase, even though there's solid domains, it is rapidly reorganizing to always be coupled. And it's that way across the entire sample. Okay? We never see any indication of diffraction from uncoupled leaflets. Okay? At a larger scale, you can also look at this. This is DPPC domains that are in a fluid phase of DOPC. And you can actually have these new dyes that you get a different, you can polarize the light and you actually get a different shift depending on if the dye is oriented or not within the leaflet. And you can look around and see the texture. Now this is 5 microns, these leaflets. And this is a picture of actually mapping in the center this sort of texture from these different orientations on a larger scale. So it's exactly a demonstration of hexagonal, where you have short-range positional order, but long-range orientational order in the system. Okay. There's some other aspects. So we look at this and we're like, oh great, we have these defects, and it looks like it maps on exactly to liquid crystals, which is nice with the lipid system, with the rich physics there. And there's another system that has some very interesting aspects as well along these lines. And that's when you have protein binding. This is a case of a lipid monolayer, and it has a lipid receptor, the native lipid receptor for cholera toxin. And cholera toxin is kind of unique because it has 5 binding pockets for the receptor on each protein. And so you can get some interesting impact when the protein binds of how it's going to reorganize the lipid layer. And what I show here on the bottom is different surface pressures, and on the left is just the monolayer without protein, and on the right is the monolayer once protein is bound. So surface pressure is 45, 30, and 20. And what you end up seeing is that as the protein starts to bind, it actually starts to distort the packing. It opens up the area per molecule. You see an increase in the area, and the lipid molecules start to tilt over. And so you get this more and more off-horizon scattering intensity. Okay? And in fact, at I guess in this case, at 30 millinewtons per meter, it looks fairly, with protein, it looks fairly similar to the monolayer without any protein at 20 millinewtons per meter. So just an increase in the area. But when you start to look at how the lipid and the receptor have to be bound in that receptor binding pocket, that tilt of the molecules is sort of like you can think of it, we're trying to relay this over to superconductors in the magnetic moment. You actually get vectors within the lipid layer. And you can start to model when these receptors are locked into the binding pocket, again there's 5 on a protein, it actually sets a vector and how the other lipids would have to assemble around to maintain the packing and the diffraction patterns that we see. And so you have things like vortices and so forth, and I'm sure if we blew this out, you'd have different sorts of defects and so forth. But these sorts of interactions that we have, and you have a mapping of different vortices around, it's a way that you might even imagine when there's a question of when the cytoskeleton is interacting with the membrane, somehow you have to have a long-range but soft interaction with these points of how the cytoskeleton, the proteins are networked into the membrane. This sort of example would be a case where you can have long-range interactions between these vortices, okay? But it would be a very soft interaction. So we're wondering if these are locations of that this sort of behavior would also happen in biological systems as well. So, and we're playing around more with diffraction and looking at the sorts of defects and things that we see. So I'm going to skip that. In my last few minutes, I wanted to get a little bit to some practical aspects and how do you make a biosensor with these sorts of systems. And the first thing is if you want to stick proteins in, you can't do that when it's right up against a surface because proteins denature. So you've got to go to something where you lift the membrane away from the surface. And just playing around with electrostatics is sufficient. You can do things that are a couple of nanometers, but we really want to have something that's 10 nanometers away from the surface. And so the way to do that is put a polymer cushion in. And many people have been looking at this. And I'll show you some examples of, I'm just going to pick out two. One is where we put a polymer against, that's covalently grafted to the lipid and make a hybrid molecule to try and lift the bilayer up. And the other is where we put down a pH-responsive polymer layer and deposit the membrane on top of that. And if we can get proteins into the membrane, it also opens up our ability to try to crystallize those proteins and use some of this diffraction to understand better structure of integral membrane proteins. And actually, let me back up for a second. Why that would be so critical, because it's not on my slide. If you go into the protein data bank, there are, I guess it's probably around 30,000, or maybe it's over 36,000 protein structures in the protein data bank. And if you look at drugs, 60% of all drugs on the market interact with integral membrane proteins. And if you can see the structure of a protein, it's much easier to design a drug to alter that protein function. So does anybody want to guess on how many of those 36,000 protein structures are integral proteins? So proteins that actually span the membrane, which most drugs interact with. Anybody going to give me a guess? 10%. So that'd be 2,000. Almost 4,000.
217.
Yeah, actually, in fact, if you go to unique ones, it's less than 200, right? So less than 200.
200 unique.
Yeah. Okay. Well, it's increased since I last looked, 200 unique. So if you have ways of actually crystallizing integral membrane proteins, this would be very powerful. So just with time, you can attach a polymer to a lipid. You can have that polymer be non-reactive or reactive, so it can actually graft to the support. You get very different structures depending on what you do. But what you actually see in this case, and I'm going to do this poorly because I'm going too quickly, is that when the end of the polymer is reactive and combined with the support, you actually get a bimodal distribution. Some of the bilayer is lifted up on the cushion of the polymer, and some of it is right up against the surface. There's a length scale here on the order of half a micron, and I think you can play around with this sort of fabricating regions that would have cushion where a protein, you could have an isolated protein on a cushion, and regions where the membrane would be right up against the support and the protein would not be functioning. So it might be a way to actually pattern the system. But I'm going to skip this because I don't have time. Okay, so here's the one I want to talk about. The pH responsive cushion, it's polyacrylic acid. You can swell the polymer or collapse it depending on pH, and it works very well. You can put a membrane down on top of this, you get a beautiful membrane. We haven't checked for sealing, but it's not too bad against the cushion. Let's see, skip this. You can pattern this system, so we can pattern the PAA, so the regions that are bright here have PAA and a membrane on them. The regions that are darker just have a membrane. You look at diffusion of the lipid layer, and the diffusion is about 1 micron per second in both regions. Or excuse me, it's about 1 micron per second on the PAA, it's about 4 microns per second, microns squared per second in the uncushioned region. So it's a little bit hindered due to the interactions with the cushion, but it's not too bad. Everything recovers. You can also change pH and look at how long it takes for this underlying responsive polymer to change, swell, and collapse. When you have a membrane there, it actually takes on the order of a couple of hours. So the membrane screening, the change in pH. But if you put a protein that actually is an ion channel within the membrane, you rapidly see height changes now. So with a grammocyte that allows protons to go rapidly through the membrane, you get a change in height of the cushion in the course of about 5 to 10 minutes. Okay? Versus hours. And then finally, let's see here. How are we going to crystallize proteins? Well, the great thing about this system is that the proteins diffuse pretty readily, and you can apply an electric field not across the membrane this way, but laterally along the membrane. And you can fabricate barriers within the membrane. You can either remove the membrane or put a PDMS barrier. And you can actually collect any proteins that you incorporate into the bilayer against that barrier. So this is, on the bottom, there's an electric field gradient laterally along the membrane. There's a barrier here. This is lipids that are charged that are collecting at the barrier. And then we put in a protein, it happens to be a nexin, and now this is the protein that's collected at the barrier. So you have a way of concentrating proteins in a specific region of the membrane that might enable you now to play around with crystallization. And that's what we're really spending a lot of time on now. And with that, I'll end. So thank you very much.
Basically, the gradient X-ray or neutron spectrum using very, very low angle instant beam, right? That means it shows, it contains a bunch of layers and very large area information, and it contains very large area information. So how can you determine the kind of protein and how much? How can you determine the amount of protein in it? Because in your skin, the protein is very small amount in it. So I guess the gradient X-ray or neutron spectrum data shows mainly some bilayer information.
So we have gotten protein diffraction, okay, from a single layer of protein, but absorbed to the membrane. So we had a receptor in the membrane. We found protein, so it wasn't integral membrane protein, but a protein that bound to the receptor, similar to the cholera toxin example. That protein formed a crystal, and we were able to measure diffraction from that. Even under the best case scenario, our diffraction resolution would be on the order of 7 to 10 angstroms. That's nothing like 1 to 2 angstroms for a really nice 3D crystal. Okay. However, if we can crystallize in 2 dimensions, we can also do electron diffraction, right? And there we can get 2 to 3 angstroms if we have nice 2-dimensional crystals in terms of resolution. So I don't think X-rays are going to be the solution for crystal structure determination, but it is an easy way to screen and do all these things and then go, okay, here's the way we want to make the sample, and now transfer that sample to an EM grid and do it with electron. now, in question to how much protein we can get in, our samples are large. So we have our substrates are 1 by 5 centimeters. Okay, we can put a barrier at one end of the sample, and if we get 0.5% protein loading, we can collect all of that protein across the entire area against the barrier. And we can look at the region where the protein's all collected and only have that be a beam that's 100 microns across. Okay. So there's a lot of things, and this is just what we're using. We can make that sample much longer, right, and collect even more protein. Okay. Proteins move fairly rapidly in the electric field, but of course there'll be limitations on if we're destroying anything by applying the sort of gradients in the electric field. I mean, there'll be problems, pluses and minuses to how we do this.
More questions? If not, then thank our speaker.